Dr. Daniel, can you share your screen? It seems the professor is absent. Oh, really? Yeah. Please move on. The next. Okay. So the next. So we go to the next. Okay. So the time is me. So okay. So we just skip here, and then if Dr. Daniel come back, we'll ask Dr. Daniel to give a talk again. So the next speaker is myself. I'm 𝗍𝗫 from Institute of Genetics and Developmental Biology, Chinese Academy of Sciences. Today I will give a talk of functional genomics and molecular design grading in soybean in our lab actually. So I would like to share my screen, right? Okay. So can you hear me, right? Can you see my screen? Yes. Okay. Okay. Thank you. So thanks a lot of that's the program committee or the organizers to okay. So give me this chance or opportunity to report our work. So our lab mainly work on the functional genomics and also do some molecular design grading. Here is my talk. So just like Dr. Daniel said that. Dr. Richard said that the soybean, during the last maybe 60 years, at least in China, not increased so much, but in Brazil it's increased a lot. But in China, didn't you see only maybe a little bit, but particularly compared with the maize, rice, and wheat. So in the last 60 years, maize, rice, and wheat, the yield has increased about 3 or 4 folds. So that's because of the Green Revolution, right? So there is a call for the Green Revolution for soybean. So in last year, we summarize of some perspective on how to get the Green Revolution for soybean. So that's our goal actually. That we want to try to get some tools or some knowledge to achieve this Green Revolution. So how we can get that in our lab? We mainly work on like this kind of strategy. So we use the genome approach. Also use a lot of germplasm to do use the different omics approaches to do the 𝗍𝗵𝗶 analysis, and then to identify or review the genetic network to control different the trees, and also we try to do some functional study to validate the genes, and then use this kind of knowledge or information to do the molecular design grading. So this is our strategy. So today I will introduce our work based on the first genomic work, also some the 𝗍𝗵𝗶 analysis for some trees, particularly to identify the genetic network to cover different trees, and then also some grading, a little bit of the grading. So for the soybean genome, the reference genome is published or released in 2010. So after that, I got back to China from Purdue. So we start to work on the soybean genome. So for soybean genome, one distinguished character is that the duplication. So at the first series, we try to understand how the duplications are rooted. So we published some papers. I don't want to introduce too much on this part today. So, but when we do the genome, you know, at that time we only use the Williams A2 as a reference to do this kind of work. But step by step, we found some questions or problem for the reference genome. You know, for the 𝗲𝗏 analysis, you can see the signal is very straight. You know, you can see here this signal not like this kind of normal signal. We think there must be some problem for the assembly because you know the Williams A2, that's the reference genome assembly, is still based on the second generation resequencing technology, so have some limitations there. So we want to improve the reference genome. One strategy is to still improve the reference genome of Williams A2. Another is to assemble a new reference genome. Then maybe we can add some more information together with the Williams A2. So later on we decide to choose the second strategy, so to construct a new reference. So we select one varieties from China, 𝗠𗲲𝗷𝗆 because it's a very famous varieties here in China. It is developed by the Institute of Crop Sciences, Chinese Academy of Agricultural Sciences in 2001. So because of it's very high that the yield or stable in different adaptation to different areas. Also, the largest that's the varieties being planted in China now. So it's very famous. So we decide to assemble the reference genome for these varieties. So benefit from the new technologies, particularly for the 𝗏𝗏 or the 𝗏𝗏nano. So with this third generation, the sequence, we got a very good quality of the reference genome because the technology has increased a lot, right? So we published this reference genome in the Science Journal of Life Sciences in 2018. The contig M50 has been 0.46 megabits, and scaffold M50 is almost half of the chromosome. But in the next year, that means in 2019, we get a new assembly pipeline can further improve the quality of the assembly. Then we decide to update the reference genome again. So this time the quality is really good. It has been reached to a golden, that's the level, because you can see the contig M50 has been half of the chromosome, and the scaffold M50 is almost the full chromosome. That means only one gap. The big gap is only one for each chromosome. So still published in the released to the Science Journal of Life Sciences. If you want to get the reference genome, also include some of the RNA-seq for different tissues, also microRNA, that's the sequencing data for different tissues. You can get from this here. So after we got the reference genome of 𝗠𗲲𝗷𝗆, here you can see correct some assembly error for the Williams A2, because you can see for the strange signal for the 𝗲𝗏 because it is assembled to the wrong loci at the chromosome 6. It should be in the chromosome 13. So you can see after recorrection, the 𝗲𝗏 signal is normal now, right? So we got the 𝗠𝗲𝗷𝗆 reference genome. Then we realized maybe one and another for the Williams A2, only one or two reference genomes still not enough to allow us to identify some important structural variations to contribute the trace variation. So there is a need for the pan genome. This is not only for soybean, for most of the crops. we can say for all crops or even plants or biology, session. So we need this kind of plant genome. So we can, for species, that's important to identify the structural variations to reflect all the genetic variations. This is not new, but how to construct a real plant genome, not a multiple individual genome, that's a challenge actually. So in this review, they propose two. That's the direction. One is use the iterative assembly this approach, but this approach has a limitation. For example, you use different genome as a reference, you can get different the integrates the genome, right? So you can see here different. Also, for some, that's the genome you can see here is artificially combined together. Actually, in the different genome, they don't have this kind of combination. So another approach is the variation graphs. So this kind of is very promising, but how to construct this kind of new genome is very challenging. So we decided to use the second approach to do this. So we first the sequencing 2,898 soybean sections based on the phylogenetic analysis of all genetic version. And then we select 26 and plus the 27 sections to do the de novo assembly of all these sections. So we used also still for the third generation sequencing technology, also use the 𝗖𝗵𝗊 and 𝗖𝗵𝗂𝗀𝗃 and to do the assembly. After assembling, we got a very good quality the assembly genomes for each of the section. You can see here the character is very good. So based on the individual assembly the genomes, we do the genome analysis to identify the core and the dispensable gene family or structure here, also for the structural variations, and then use the core and dispensable gene family and structural variations. Then we to construct the graph-based plant genome. This is the pipeline here, and then after we got this, we use this almost 3,000 the receiving sessions to map to the new genome to identify the structural variation for each accession. Also, we identify some new structural variations here. Use the structural variation, we can do the same work as the genetic variation. Previously used the second generation resequencing data, for example, SMP or small indels. Here, the same thing is only for the structural variation. So we can identify some structural variations that contribute to some trait variation. Here is a sample. You can, for the lucerase, this trait you can here, we use the structural variation that's as the to do the 𝗲𝗏𝗊𝗂𝗀. You can see here in the chromosome 15 have a very strong signal here. We found that the absence and the presence of this gene is associated with the trace variation here. And another sample is you can see here for this iron deficiency chlorosis. This is a very important trace, you know, right? So there is a QTL previously identified. In this QTL, we found 1.4 kb in there actually associated for the expression of this gene also contributes to this trait. So you will see the results or the information for this plant genome will give you a lot of information for the structural variation. You can do some analysis in future if you need it. Okay, so actually all the data has been released to the database. identify the trees that associate with the agronomic trees, or also related to the domestication improvement. For the soybean breeding, it's the domestication and then introduced to different area, right? I don't want to repeat this kind of history. So to do this, actually, several years ago, we been sequencing about 300 soybeans, including the wild soybean, landrace, and 𝗏𝗏𝗏. We also, you will know, right? So for the genetic diversity during domestication and improvement has been decreased a lot. That means the genetic diversity in the 𝗏𝗏𝗏 have very low now. It's very limited to also have some limitation for the further improvement, right? If we want to further improve some trees, particularly for the stresses. Then we screen for the select the sweeps that's associated with domestication and improvement. We identify 230, that's the sweeps. And actually, this kind of analysis cannot give you the information how these select sweeps contribute the trees. Then, to answer this question, we phenotype all the domestication and the improvement trees for the accessions, and then we do the 𝗲𝗏𝗁𝗂𝗀𝗃 analysis, and also integrate some progress 𝗀𝗃𝗷 analysis, and then combine all this information together with the selected sweeps. We can answer this signal is associated with the trace of oil percent, and this selection is associated with the seed weight, also some. So that means for this part we can answer for the selection signals the associated weight with different trees, right? So use this kind of approaches. We also clone and some important genes, for example, that yield related, collaborate with Professor Fan Jiangkong and Professor 𝗵𝗶𝗰 this group to clone the flowering time related genes and the photosynthesis related genes and also some stress related genes, right? This is collaborated with Professor 𝗲𝗁𝗁𝗀 actually in the Zhejiang University. This weight 10 is very interesting actually. Country the seed side also associated with the oil and protein that content actually also selected during the domestication. It's a very interesting story. So when we get the different genes, actually we found just like the weight 10, actually one gene not only contribute to one trait. So this, if one gene can affect different traits, usually can make the trees of different traits to be treated off. That's also important for our breeding, right? So you need to treat off different trees to get a balance there. How to get this kind of the trade-off for the trees? So to answer this question, we sequencing on the phenotype the 800 soybean sections and do after we phenotype them, we do find that the 84 trees have some association among each other. Maybe the correlation may be strong or weak, but however they have some associations, correlations. For example, if you want to change this trait, usually another trait here that showed a very high correlated should be changed, right? So if you want to only change one trait for each session, how to break this kind of trade-off? That's the question, also a challenge to answer this. So we do the 𝗲𝗏𝗁𝗂𝗀𝗃 analysis to identify the loci controlling each of the trees. To do the 𝗲𝗏𝗁𝗂 analysis for this. So for individual trees, we got the loci that contribute to the trees, and then when we put all the 𝗀𝗃𝗷 or the loci control different traits to the 20 chromosomes, we found some that's the pattern. For example, for this loci only contribute to one trait, for example, trait 20, but for some that loci can contribute to different traits, So how to connect this kind of knob, or we call it knob, and to the correlation of different traits, and then we use the RD to connect the different loci. So we get this genetic. To get this genetic networks. This network can answer some questions of the correlation of different trees. Actually, this genetic network have several components. One component is the trait. The second component is the loci to associate with this trait. Use a line to connect them. The third is the RD, or the RD of different loci. If they have some, if they have the correlation, they have the correlation, they will be highly connected. So this can give you some information for how to break the trade-off. For example, these three traits have very strong correlation. So if you want to change all the three traits together, so the use of this 𝗏G is very important. Selection of this 𝗏D is very important. But if you want to break this trade-off, you only want to change this trait or improve this trait. Only can use the loci control this specifically to control this trait, right? So we use this approach. We do the molecular breeding. We indeed get some new varieties. For example, here we use two parents with not so high protein content, but after we combine, we categorize all the loci together because the new variety has the high protein content and also the yield increase. So that's all my talk today. So I would like to take this opportunity to thank all my collaborators, also the lab members in our lab, also the fundings that support us. Thank you so much. So, okay, I think still, I'm sorry, we don't have the time for the questions. So, I'm sorry, Dr. Daniel Calderi, are you here?
Yes, I am here.
Okay, I'm sorry. That's how I didn't get the information. Didn't hear from you. So now we go back. So could you give the talk now?
Yeah, I can start.
Yes. Can I start? First, I introduce a little bit. So the next speaker is Dr. Daniel Calderi, and then the topic is solving as a new and promising crops for southern Chile. So thank you so much. Please.
Sorry everyone, because there was a confusion. We tried from Chile. We tried to connect, and some problems happened. But anyway, I will start the talk. I will share the screen. Well, first of all, I wish to thank the organization of this international symposium and everyone who is watching this presentation and the symposium. Well, soybean is a very new crop in Chile. Actually, we are trying to understand the response and the yield and quality, grain quality of this crop, because about or around 2050. You maybe several of you know this figure where you can see the present trends or almost present but trends in grain yield of different crops, most of them cereals, but also soybean. The projection, if no change happens, and the projection to supply the food demand in the future. We have, as the title of this picture, a big challenge ahead. Also, climate change is challenging us to try to adapt the crops to higher temperatures and also different heat waves that will affect green yield across different crops. Well, these are the projections, different estimations of the increasing temperature. More unpredictable is rainfall, as you know, and several areas of the planet are endangered by the soil degradation. In this context, we are seeing an increasing change in the regular source of food of the humanity. In this context, plant-based food are very interesting and growing, which will demand more plant proteins. And as you know, soybean is a very important source of plant protein. Looking at our country, in Chile, salmon industry is very important. Actually, Chile is the second salmon exporter in the world, which is demanding plant protein and plant oil also. Because of that, soybean could be a very useful crop for Chilean farmers to provide to this industry. In the present time, this industry is supplied mainly by soybean produced in Argentina, our neighbor country, and most of this soybean is grown in the central area of Argentina using transgenic soybean, which could be a problem for the market of the salmon industry. Soybean has three main nodes of production in the Americas. North America, mainly the United States, and in South America, two nodes. One is Brazil, which is the highest soybean producer in the world, and the second one is Argentina, which is the main exporter in the world. But in Chile, here is Chile, a very long and narrow country. There are, as I say, very few examples could be found of soybean production. But in the new context, it's a very promising vegetation and temperature is very good in our environment. Actually, the coefficient, the quotient, quotient between radiation and temperature is very high compared with many other places in the world. Evapotranspiration is low, and the photoperiod, as you know, is increasing southern. Just to show you some evidences of the very high greening potential of our environment in southern Chile. This is an experiment from a few years ago, or several years ago, where double haploid lines with cultivars released in Chile were evaluated, and as you can see, the experiment reached 16 tons per hectare. Also, in a legume crop as lentil, the potential is also very, very high. These experiments were carried out at potential conditions, both in wheat and also in lentil. Almost 8 tons per hectare in lentil. This is a comparison in this paper between Valdivia, our city, and Roseworthy in southern Australia. Just to put your attention in this, precipitation is in blue, and evapotranspiration, potential evapotranspiration is in red. You can see that during the crop cycle of lentil, almost most of the crop cycle is plenty of water. In addition, our soils has a water availability of 200 millimeters, more or less, and as far as 1 meter, 1.2 meters in depth. Very contrasting with southern Australia. Okay, I will show you the experiments carried out last season in two locations in southern Chile. Los Angeles, which is at 37 degrees of south latitude, and Valdivia, our city, which is almost 40 degrees of south latitude. Sowing dates were November 15 in Los Angeles, northern than Valdivia, and in Valdivia, two sowing dates, November 11 and December 9. The cultivars used were Pompey, Capral, and 𝗖𝗵𝗶. I will focus my presentation in these two cultivars, Pompey and Capral. Well, plant density was 33 plants per square meter. Here is the experimental design, 3 reps. The experiment were in Valdivia were fertilized with nitrogen because we were not sure about symbiotic relationship with this crop, because we don't have any previous data or background. On the contrary, in Los Angeles, where the rhizobium nodulation was used in the the sowing day, as I say, was around November 15, and similar in Valdivia. These were the crop cycles of sowing, the first sowing and the second one. The phenology of this crop as it is shown in this picture. The first sowing, November 11, which is almost May 11 in the North Hemisphere, completely the crops. But the second sowing day, at the beginning of December, was impossible to complete the crop cycle because temperatures are very low at that time, and definitively we can sow this crop in December. These were the results of the experiment. Grain yield reached between 3.5 and almost 4 tons per hectare of 𝗿𝗶𝗰 and 𝗀𝗵𝗋 in the northern location in Los Angeles. But in Valdivia, grain yield reached 6 tons per hectare in 𝗿𝗶𝗰 and 4.4 tons per hectare in 𝗀𝗵𝗋. Interesting that the protein concentration was 38% in Los Angeles and 41-42% in Valdivia. Meanwhile, oil concentration was around 23% in Los Angeles and lower in Valdivia, almost 20%, between 19 and 20%. But these values, grain yield and protein concentration, are very, very good for the first trial of this new crop. As always, which is very usual in crops, grain yield was more associated with grain number than with thousand grain weight. We know that we have very few data actually, but in this set of data, we confirm that grain number is the main driver of grain yield also in southern Chile for soybean. We also found a trade-off between oil and protein concentration, which is generally reported in literature. As I say, we have very few data. Actually, this seems to be only 2 groups of data, but is the only information we have available today. We hope to increase this information in the next years, the present year and next years. This is radiation interception just in Valdivia of 𝗿𝗶𝗰 and 𝗀𝗵𝗋. And the important thing about this is the critical period. As most of the grain crops, there is a phase where grain number and grain yield also is defined. This period, this window, this crop season window is very important to understand intersection in Valdivia of these two cultivars, you can see that they reach the critical window almost fully interception of radiation. And full interception of radiation started before the critical window, which is very promising for our understanding of the high yielding reached by, especially by Pompeii, during the first sowing season. These are the environmental conditions of Valdivia and Los Angeles about temperature, maximum, minimum, radiation, and cumulative rainfall. These last years were very dry for southern Chile, especially in Los Angeles. You can see the gap, the very big gap. But this is unusual in Valdivia, where most of the seasons precipitation is over, or at least close to evapotranspiration, potential evapotranspiration. Here is RC, and R5 is almost here. And you can see the critical period, or the critical window for grain number determination. We think, as in other crops, that this critical window is very favorable in southern Chile. I showed you the different seasons of soybean in different locations. In North Hemisphere, in the South Hemisphere, Brazil, Argentina, the crop sites thrive in the fair experiment, but we are planning to move the sowing day in advance to improve grain yield of this crop. Actually, we are trying sowing dates, but in the new season 2021, we already sown earlier than the first experiment, and we expect higher grain yield. This is the presentation. We only have one season, but this give us the chance. This allow us to say that southern Chile is a good location or good area for soybean, and it's very promising, especially for these cultivars which are not transgenic. And the lack of transgenic cultivars are very important for salmon industry in our country. Again, thank you very much for your attention. Thanks.
Thank you so much for Dr. Daniel. So Dr. Daniel gave us a very good talk about the very comprehensive, that's the trail for the how to plant the soybean in different seasons at a different location. So give us the guides to plant soybean in the southern till in different locations at the right season. So it's very, very promising. We are looking forward to get the further that maybe information later on to learn how to plant the soybean in till. So thank you very much. So for the timing issue, so we switch to the next speaker. So the next speaker is Professor.
Thank you.
Thank you. So Professor 𝗖𝗵𝗶H，然后from the University of Valdivia de Mato Grosso from Brazil. The topic is new technologies for sustainable management of the soybean crops. So welcome. So thanks.
Boa noite.
Yeah, we see the screen now.
Okay, good evening. He's just preparing the presentation. Okay.
Ok. Então, meu nome é Cassiano.
I'm Cassiano, and professor in the University of Mato Grosso in the northern region.
Então, sou engenheiro agrônomo, né? Fiz mestrado em fitotecnia e doutorado em fitotecnia também.
I'm originally an agronomy engineer. My Master of Science was in phytotechnics, and my PhD was as well in phytotechnics.
Hoje eu vou falar para vocês sobre 3 patentes que eu desenvolvi durante toda a minha carreira profissional.
Today I'm going to talk about 3 patents I developed during all my career, my professional career.
Inicialmente, né, a primeira patente minha foi com a aplicação de própolis na agricultura.
So first, my first patent was about putting propolis, the application of propolis in agriculture.
Tudo começou um dia que eu estava gripado, né? Eu comecei a tomar própolis, um extrato de própolis, né? E passou ali 2 dias e eu melhorei. E aí eu tive a ideia de fazer aplicação em culturas agrícolas.
So everything started when I faced a very harsh flu, and in 2 days I improved really well, like I was really cured. So I started to think, okay, why not applying propolis in the crops?
Então, para falar um pouquinho sobre a própolis, né? Mostrando aqui uma própolis que a própolis normalmente ela é retirada das caixas, das colmeias, né? De forma bruta, e depois a gente vai fazer o extrato. Então, só mostrando na figura aí.
So you can see in the pictures that I take the brute substance of propolis and then I will prepare the extract of propolis. And it's a special substance against insects, bacteria and fungi.
Então tá mostrando aí, né, a própolis, a gente retira essa camada de própolis que está na colmeia ali, né, mostrar um pouquinho aqui também, né, a própolis então serve para as colmeias, para a proteção das colmeias, principalmente contra fungos e bactérias.
So the first picture of the previous slide, I was talking about the propolis, and now you can see the details of the bees sealing the beehives. So the box are sealed with propolis. So the substance you can see in the walls of the boxes here, yeah, especially in the corner of the, in the right corner, as you can see in the circle.
Bom, aqui mostrando, né, a nossa apicultura, né, e aí os produtos apícolas, né, então a resina aí que é essa própolis bruta.
So you can see our apiculture and all the resin and the substance of the brute propolis. So you can see the honey and also the propolis, but in its brute characteristics. So you can see in the picture above the apiary and in the picture below the propolis extract and the beehive.
A partir da própolis bruta a gente faz o extrato, que é o que a gente está mostrando nessa figura, né? Então o extrato a gente coloca a própolis bruta dentro do álcool, né, e deixa por um período até produzir o extrato de própolis.
So you can see from the picture that we extract the propolis and we leave it in immersed in alcohol, and it's from this alcohol that the root propolis is immersed that we take the extract of propolis.
Então, inicialmente a gente começou a trabalhar com a aplicação de própolis para controlar doenças, né, fungos presentes na cultura do cafeeiro, né?
So the first type of crops that I have tried to cure the diseases was coffee plantations, southern Minas, to combat and actually also to control the disease in coffee plants. So you can see, as you can see, the first tribe was in coffee plantations.
Então, estão aí, né, as duas doenças aí do café, né, que é a ferrugem e a 𝗟𝘁𝗂𝘀.
So the main diseases that you can find in coffee is the brown eye spot and the coffee rust. So these are some of the examples of the diseases that I wanted to combat or to control in the coffee plantations.
E aqui é um dos resultados, não vou falar muito deles.
Okay, so you can see the results. I will not explain a lot about the results, but they are very productive. You can see that there was a high improvement curing the coffee diseases. So here you can see the one graph about the brown eye spot, the coffee, and then you can see the other type of diseases. And propolis is a natural substance, and it improved a lot in curing coffee diseases.
Então nós verificamos então que a própolis tem certo controle, né, tanto sobre a ferrugem do cafeiro quanto sobre a 𝗟𝘁𝗂𝘀.
So we could verify that it was an effective medicine to cure the brown eye spot and the rust in the coffee. We have proof this in the graphs.
Efeito é em torno de 50%.
The effect is around 50%.
Bom, aí depois eu comecei a trabalhar também com a aplicação de própolis então, na cultura do feijoeiro e da cultura da soja.
Then I started to apply propolis in the cultures of black type, valente bean, and soybean. So that is my experience with soybean as well.
Bom, aqui mostrando novamente, né, a própolis, né, própolis bruta, e aqui a preparação do extrato, né? Então pega a própolis bruta, dilui no álcool, dilui na água, depois a gente faz a pulverização na cultura. Aí mostrando uma planta de feijão, né, que a gente aplicou a própolis.
You can see in the pictures all the circle and the process, how to do the extract, and also the solution, the liquid that's going to be sprayed in the crops. In the first picture, you can see the raw propolis, and then the propolis immersed in alcohol. After immersed in alcohol, you can see the solution that's also mixed with water. It's a very milky solution, and this milky solution is a solution that's sprayed in the plantation, and after spraying the plantation, you can see how the leaves are left with the solution.
O mais interessante é que a própolis, por ter cera, né, então ela gruda na planta e fica um cheiro muito bom durante aí aproximadamente quase um mês da cultura, né?
The most interesting part is that because propolis has also inside the composition wax, and the wax is something that gets attached to the leaves, and in the cultures you can see through the plantations all the propolis, and you can feel an amazing and pleasant smell of propolis that stands for about 1 month in the plantation.
Aqui só mostrando um detalhe, né, da própolis já diluída em água para aplicação.
So I'm just showing the detail of the solution just ready to apply in the crops.
Nós colocando então no pulverizador, né?
So we are putting this in the sprayer to apply to the crops. As you can see, the student who is adding to the sprayer the solution.
E aqui o experimento, né, onde a gente mostra as parcelas, né, onde foi aplicado própolis, as testemunhas e tudo mais do experimento em feijão.
So this is the actual experiment. You can see the parts of the crops that we applied the propolis, and you also can see that it's divided, and in each part you have a core sample, and then you've got another samples that we applied the propolis.
Aqui o experimento feijoeiro, também mostrando o experimento grande que nós fizemos aqui em Sinop.
So this is a very huge experiment we did with the black type Valenti bean, and it's a big plantation that we just did the experiment in Sinop, the northern part of Mato Grosso.
Aqui os alunos fazendo as avaliações que nós fizemos durante toda a cultura do feijoeiro, né?
So you can see my students making evaluations, checking the efficiency of propolis in beans. So this is the black type Valenti beans.
Aqui só mostrando detalhes, né, da aplicação da própolis, né, da cauda própolis, né, já diluído em água, né, na cultura do feijão.
You can see actually the propolis in its liquid version and the solution, the milky solution, as I told the process before, and this is the way the leaves are left after spray.
Aqui mostrando também, né, alguns trabalhos que a gente já fez, né, na cultura dos citros, do café, do feijão.
Now you can also see that I have at the moment I just have applied first in an orange tree, citrus, then in a coffee plant, and then in the black type Valenti bean.
Aqui a aplicação sendo feita no campo, né?
So this is the moment we are actually spraying this in the field.
Um outro experimento que nós fizemos com feijão dentro da UFMT, da nossa universidade aqui.
So this is another experiment we have done in UFMT here in the university.
E aqui os alunos avaliando nossos experimentos aí com próprios da cultura aí do feijão também, né?
So you can see all the students, they are also checking and verifying, so they are making evaluation of the efficiency of applying propolis in the crops.
Um dos resultados que a gente achou bastante interessante foi que a aplicação da própolis devido à presença de ceras reduziu a perda de água da cultura.
A very interesting result that we found very important is actually because propolis has wax in it, so the bean leaves, they became kind of water resistant and the leaves could retain water. So in times of drought, it's a special thing to apply to the crops because the leaves retain the water with the propolis application.
Então verificamos aí aumento na quantidade de água foliar, produtividade, né? E controle de doenças também, principalmente a ferrugem e a mancha-alvo.
So one of the other, there is a Chinese sound, I beg your pardon. So one of the graphics, here you can show some results with the application of propolis. So you can realize the increases of productivity and the drought tolerance. And you can see that it increased a lot the capacity of the leaves to retain the water.
Agora os experimentos com soja, né, que nós fizemos aí a aplicação de próprios na cultura da soja.
Then these are the experiments actually with the soybean. So you can see here that we are going to start to talk about the soybean crops.
Aqui só mostrando, né, e um detalhe aí, né, na parte superior do vídeo, onde foi aplicado o própolis já próximo à colheita, e na parte inferior, onde não foi aplicado o própolis, a testemunha. Então a gente verifica aí que a própolis conseguiu manter mais as folhas na planta, devido principalmente ao controle da ferrugem.
You can see that in the picture above, it's the soybean crops, wax that actually we had applied propolis, so it's richer, yeah. And actually, the other in the below part, you can see that our core sample, the core sample which was not applied the propolis, it's kind of yellowishy and paler.
Bom, e aí nós verificamos então da cultura aí também o controle da mancha-alvo, né? E mais uma vez verificamos aí que a própolis manteve também, reduziu a perda de água da cultura da soja também.
So we first checked that we could avoid the brown eye spot, and also another time we could prove again that the application of propolis in crops helps to improve not losing water in the leaves of soybeans as well.
Bom, aí eu expliquei a primeira patente minha, né? Agora eu vou para a segunda patente, também desenvolvida por mim durante o meu mestrado.
So I just explained my first patent, and this is the second one that I developed during my Master of Sciences about biodegradable beeswax tubes.
Aí essa segunda patente foi relacionada então ao uso na verdade seria a substituição do plástico em várias etapas da agricultura. A gente sabe que o plástico é um grande poluente ambiental, né? É o maior poluente dos lixões, né? E aí a gente substituiria então o plástico por ceras na produção de mudas nas culturas perenes, né?
So the intention, it was actually to replace plastic in the plantations. We know that most of our pollution is plastic. So my idea was to replace the plastic tubes by wax tubes and actually reducing pollution. Yeah.
Aí o que nós fizemos, né? A gente derrete a cera, né? Tem um molde, a gente mergulha o molde na cera, tira o molde, aí a cera se seca, né? E a gente produz o tubete.
So here you can see there are molds, and with the molds we dip them in the melted wax, and with the wax we make the tubes. So with these little tubes, these are the ones that we patent the process as well to produce tubes made of wax and not plastic.
E aí está o resultado da nossa fabricação, né? Os tubetes, né? A gente faz com várias espessuras, né?
So these are the results of our industry. We are actually doing this in scale. So you can see the wax container, and this is also a process that we made the second patent.
Aqui então a produção das mudas, né? Aqui é a muda de café, já com 2 meses aproximadamente, né? Sendo produzida aí no tubete de cera, né?
Sim. You can see that these are coffee seedlings, and they are already 2 old. So you can see that the sprouts are 2 months old in the wax tube.
Só mais um detalhe aí mostrando, né? Então a produção, né?
So another detail showing the production. So you can see the sprouts. This is a 2-month-old sprouts in the wax tube.
E aqui só mostrar então o que o produtor pode fazer. Ele pode plantar diretamente essa planta, né, com um tubete. O tubete derrete, as raízes vão quebrando ele e vão saindo no solo. Ou então o produtor pode tirar o tubete, derreter e refazer o tubete, sem contaminar o meio ambiente.
The producer has 2 options. The first option, he can plant the sprouts with the wax tube, and the wax tube will melt into the soil, and in the soil the roots will spread out, and it's okay because the soil will absorb the wax. The second option is actually to reuse the wax and make new tubes again, so you can reproduce and everything will be fine again. Yeah, and it's a more sustainable part in agriculture.
Bom, aí eu terceira patente agora, né?
So this is my third patent now that I'm going to explain the plant root stimulants.
Então, para essa terceira patente, eu já desenvolvi ela aqui no Mato Grosso, uma patente da UFMT, né? E a ideia surgiu porque normalmente as culturas agrícolas, né, o pessoal faz aí os tratamentos de semente, né, na parte externa, no cariópsis da semente, né, parte externa aí da casca da semente.
So first, the idea is that the traditional way to stimulate the plant root is only facing not the external part. So my idea was not doing the traditional way with chemical products, but actually using beneficial bacteria to the plants inside and not in the outside.
Nessa época eu trabalhava com experimentos com inoculação. Então, só mostrando aqui, né, quando a gente fazia os tratamentos sem inoculação e com inoculação. A inoculação é a colocação das bactérias na parte externa das sementes, né?
Externa ou interna inoculação?
Não, normalmente é na parte externa, que é feito normalmente no campo.
Okay, so this time I used to work with inoculation. The traditional way is doing the external part of the bean. What I have done, which is different, is actually inoculating the bacteria inside the bean. So you can see the not inoculated plants in your left side and de-inoculated plants in the right side, and it's a huge difference because the not inoculated is real pale and real yellowish, and de-inoculated is real green and vigor, a vigorous crop.
Sim, porém, muitas vezes no campo, o que acontece? O excesso de chuvas, principalmente aqui no Mato Grosso, acaba lavando essas bactérias.
Okay, one of the difference of my process is that here in Mato Grosso it rains a lot. So in the traditional way, when you used chemicals in the external part of the bean, what happened with the rain? It washed the chemicals. What happens with my method is actually it's a natural, a beneficial bacteria, and this bacteria inside doesn't wash with the rain, so it can rain a lot because in Mato Grosso it rains a lot. So raining a lot in Mato Grosso, with my process, there is no problem to the soybeans production.
Então, qual foi a ideia da nossa patente? Foi fazer a inoculação interna da semente, dentro da semente, tanto através da aplicação líquida quanto com a colocação de cápsulas no meio da semente, entre os cotilédones.
So the idea that we had got the patent is actually that we inoculated the liquid inside the bean. We can inoculate the liquid, or we can inoculate some capsules in the bean. So this is the idea that we have the patent.
Então aqui a gente está mostrando, né, vários pontos que a gente pode colocar ou a inoculação ou a cápsula, né?
So here I'm showing to you the points that you actually put the liquid or the capsule. So there is a cotyledone, and there are some special points where you can put the liquid or you can inoculate the capsules.
Bom, aí nós desenvolvemos então até hoje 2 experimentos, né? E verificamos então que realmente a aplicação do inoculante dentro da planta, dentro da semente, possui vantagens, né? Porque a semente, o inoculante, né, a bactéria já está dentro da cultura, né? Ela não vai se perder no solo.
So we verified with our technique that inside the bean, we don't lose the bacteria because the bacteria is inside there. So you can see that it's much more beneficial. And you can see here the 2 experiments, one with some samples yeah, about the seed, the soil, the differences in the leaves. And then you can see another experiment with the inoculation. Also, the production of soybean is more with the inoculated plant, and then it's better than the inoculated than the not inoculated.
Até mostrando aqui, né, que a planta onde foi interno aqui, né, o que acontece? Vai produzir mais nódulos de bactérias na raiz, e aí a planta tem um desenvolvimento melhor e maior assimilação de nitrogênio, né, que a bactéria, né, absorve mais nitrogênio.
When I inoculated inside the bean, the roots of the plant has more nodules and also more nitrogen. So it's better because the root is full of nodules and full of nitrogen, all in a natural way.
Aqui só mostrando mais, né, dos experimentos que foi com a cultura do feijão também.
So I'm just showing the experiment with the Valente black type bean as well. And so this is a showcase.
E aqui só mostrando alguns resultados, né, que nós verificamos então o maior desenvolvimento da cultura, né, com aplicação então dentro da semente, né, foram os melhores tratamentos que a gente verificou.
You can see in the graph some results of expressive improvement using the inoculation inside the beans. So we have proved that inoculated inside the beans, it's better to produce more beans, and it's better also for the plants, the quality of the plants.
Bom, e uma coisa, esse tratamento seria um tratamento novo, né, onde talvez toda essa questão, toda a indústria do tratamento de sementes, se fosse adotada, né, teria que ser alterado, né?
So, just to show, it's also so brand new that all the industry in inoculation would have to be changed. So it's a very important thing to think about agriculture in a different way and also in a sustainable way.
Bom, aqui está o meu contato, né? E eu agradeço a todos e pelo convite aí, também agradeço pelo convite da oportunidade de poder estar apresentando o trabalho.
So here is my contact, my email, my WhatsApp, and I would like you to be in contact because I'm available to share all my patents, techniques, and discoveries in agriculture. Thank you very much, everything. And thank you for the invitation as well.
Okay, thanks Professor Cassiano and Elisa. So Professor Cassiano introduced for the sustainable management of soybean crops by the propolis production and vaccine tubes, also some inside the inoculation of the bacteria. So yeah, it's a very different way to for the management of the crops. So because of time issue, so we switch to for this question you can leave the message in the chat box. We switch to thanks, and then we switch to the next speaker. The next speaker is Dr. 𝗪𝗁𝗂𝗀𝗰, and his topic is soybean breeding and the seeds industry in Argentina. So welcome.
Hi.
I'm sorry. Yeah.
Thank you very much.
Thank you.
Let me try to project the presentation. Let me know.
Yeah, you can share the screen. Oh yeah.
I'm sharing.
Yeah.
Do you see?
Yeah, I can see.
Okay. Good morning, China, and good evening, South America. I am 𝗪𝗁𝗂𝗀𝗰 from Argentina. I work at the National Institute of Agricultural Technology and the Research Council of Argentina, CONICET, as well. First, I would like to thank the organizer for the invitation to participate of this meeting and the possibility of sharing with you some experiences of our country related to the soybean production and seed industry in Argentina. First, it's not moving. First, we are going to contextualize current and future world scenarios in terms of climate, yield, and population evolution. After that, we are going to present soybean production, processing, and utilization in Argentina. Then we are going to address soybean and agri-food research defining quality. We are going to discuss very briefly about some factors determining seed quality through a case of a study, and of course, finally, we are going to give some perspective. So, to start, the world is experiencing warming, and this is nothing new. Since the beginning of the Industrial Revolution, the temperature has been increasing steadily all around the world. And what you are seeing here is a line plot showing the early temperature anomalies from 1880 to 2014 as recorded by 4 different institutions. You can see here that all highlight the rapid warming in the past few decades, and all highlight or shows the past decade as the warmest. What impacts is not the climate change per se, but the precipitate of the change. Besides, if we analyze the pattern of rainfall distribution in the last 100 years, this is how models have seen the mentioned distribution. This data corresponds to South America and is part of the IPCC report elaborated in 2014. But what actually had happened is what this black trend line shows, which means that no model has been able to simulate well what has happened with precipitation during the last 100 years, at least in South America. Of course, this great interannual variability in precipitation then impacts the quality of the projection, increasing the uncertainty of the expected changes. And in these figures, recently also presented by Dr. Calderini, we can see, or what is represented here is the rate of increase in productivity, that is yield per hectare per year. And it is representing here the 4 major crops, in black corn, in blue rice, in pink wheat, and in green soybean. Here in dotted lines, what actually is represented is the real yield increases. In the solid lines that follow dotted one, what is representing is the estimation of yield achievement with current yield increases. But what caught the attention is this difference between the solid lines and the dashed ones. These dashed lines, as Dr. Calderini mentioned, what are showing are the yield improvement that are required each year to double food production supply by 2050 to meet the challenges of the growing population without bringing additional land use under cultivation. So, it emerged from this figure that yield trends will be insufficient to double global crop production by 2050. And we mention this 2050 because, at least to take into account, we are nowadays currently more than 7,000 million people. And this is expected to rise by 34% by the 2050, where we are expected to be more than 9,000 million people. So, this is important to take into account because with the current population, it is estimated that there are now 800 million undernourished people. That means that the diet in terms of energy life. And the highest number of people in this condition is located in the South Asia region in the world. And the highest percentage of people undernourished or poorly fed people is located in the sub-Saharan Africa region. So it is documented that the absence of an adequate nutrition is due mainly to the lack of access to food rather than reduction in the sales of foods. But on the other side of the same coin, it is also well documented that as the purchasing power increases or grows, the amount of meat and calories consumed also grows. And the functions that relates both variables are very similar around the world, as you can see in this graphic, with some exceptions. For example, in the first graph, Asia, which exhibits lower rates of meat consumption, and in the second graph, China, which exhibits lower rates of empty calories consumption as the purchasing power increase. Empty calories are those not accompanied by protein and vitamins. So based in this information, studies estimated that the increase in the purchasing power of the population will result in an increase of up 42% of the annual meal consumption of meat per individual. Also will increase of around 11% of the calories demanded daily for consumption per individual. So it emerged a key question on how can we satisfy demands that are estimated to be bigger than current ones. In this regard, Argentina has a great opportunity, considering that we are the third world soybean producer after US and Brazil. As you can see in this first graph, the soybean cropping area in our country has been increasing steadily since the late 90s, coinciding this with the introduction of the commercial use of GMO, and resulted in 17 million hectares in the last growing season, which represent around 140% of increase respect to the 7 million hectares sown in the growing season 97-98. In the same period, the physical grain production has also increased of around 132%, passing from 19 to 45 million tons in the last growing season. So you may be wondering where or how is this soybean production distributed in our country, and this is shown in this map that you are seeing in the slide. So we need to take into account that soybean crops covers a wide ecological area in our country that goes from latitude 23 to latitude 39. But with more than 70% of the production, the rolling pampas is the main producing area. In this area, different maturity groups ranging from groups 3 to 6 find suitable environmental conditions to grow in a very broad window of planting dates that go from September to February. Besides, you may be also wondering who is leading the soybean breeding in Argentina. And the soybean, but also for all the major other grain crops, the private seeds companies are leading the breeding in our country, as illustrated by these blue bars in this graph. And as you can see here, from 2000 to 2020, there are only 2 public programs, breeding programs in our country. one located at Tucumán province, that is 𝗵𝗶𝗂𝗀𝗰𝗋 experimental station, and the other one is located in the National Institute of Agricultural Technology, that is spread all across the soybean region, all across Argentina. The other 15 breeding programs are private, basically of local development. And a special mention deserves Don Mario Company, which in the last 10 years has taken the leadership in a very significant way. And this graphic at the bottom of the slide is only to commend you that sadly the breeding programs in our country are facing a big problem since a long time ago regarding the low percentage of legal seed sown over the total sown. In green bars are represented the total area sown with soybean, and yellow bars represents the area that is sown with the legal seeds. So the difference between both bars gave us these curves, and on average 30% of the area is sown with the legal seeds, which of course reduces investment substantially and slows down companies in the launch of new varieties. But despite this discouraging context, soybean breeders have been very active in the development of new varieties, as is represented here in this graph. Sorry, I cannot move the pointer, but what is represented here in this bar graph is the evolution of the number of new varieties that are officially registered each year in Argentina since 1982. And you can see here that in 2020 there are 40 new varieties officially released. So what type of soybean do we sow in Argentina? With more or around 17 million hectares, which represent 13% of the global transgenic area, Argentina is positioned as the third largest producer of GMO or transgenics in the world after States and Brazil. And we can say that nowadays the adoption of this technology has achieved a roof, not only for soybean represented with the green bars, but also for cotton represented in blue bars and for corn too represented in orange bar in this graphic. And some factors that have helped to this very fast adoption in transgenics in our country that can be mentioned are the lower cost, the greater flexibility in cross management, decreases in the use of insecticides, and the higher yields and better quality. So I'm going fast from across this slide because something was told by Carla yesterday from our country.